Biostatistics, measure of central tendency, dispersion, and correlation. The branch of mathematics dealing with the collection, organization, analysis, interpretation, and presentation of the data. Statistics is the form of mathematical analysis which uses the quantified model. Statistics studies methodologies to gather, review, analyze, and draw the conclusion from the data. Some statistics is termed used to summarize a process that analyze the characteristic data. Okay. Statistical methods which analyze the large volumes of data and their properties. These statistics is used in the various disciplines such as psychology, business, physical and social sciences, and some economical also, humanities, government, manufacturing, statistical data. These are gathered. By using a sample procedure or other method, two types of statistical methods are used to analyze the data. The descriptive statistics is used to data from a sample to the interfacial statistics. The biostatistics is the application of the statistics which has a wide range of topics in biology. And in this biostatistics, especially the biological experiments, medicines, pharmacy, agriculture, fishery, and collection of data, summarization of the data, analysis of data from the biostatics and genetics. In the biostatical modeling forms, the important part of the numerous modern biological theories are occurred. In the general studies, science its beginning which is used to statistical concept to understand the experimental results. Some genetic sciences are advanced with the development of the methods and tools. In these biostatics and genetics, the Gregor Mendel was started the genetic studies which is used to investigate the genetic segregation in the human beings and the organisms. Solving the difference which allowed to define the concept of population is called genetics. Biostatics and medicine. Statistical concepts are present in the clinical types and epidemiological studies. These fields have their own history of biostatics development. And where is come to the frequency distribution, the each entry in the table of the frequency is displays the variant outcomes of the sample. Frequency distribution 1 minute. In this frequency distribution, we are going to discuss about the types. First of all, for what is the frequency distribution? It is a systemic presentation of the data for investigation of the sample. The frequency distribution is two types. They are grouped frequency distribution and ungrouped frequency distribution. Under this frequency distribution, the percentage distribution and graphical presentation, bivariate, multivariate frequency distributions also given. Measurement of the central tendency. This central tendency is measured by the mean, median, and mode. Okay. Mean means the sum of the average. Median means middle of the average. Mode means repeated variables which are repeated in the measure of dispersion. This measure of dispersion is also called as variability. The extent to which distribution is squeezed for measure of statistical dispersion. A measure of statistical dispersion is non-negative real numbers, that is 0. In this dispersion, the examples of the dispersion measure is standard deviation, interquartile range, mean absolute difference, median absolute deviation, average absolute deviation, distance standard deviation, coefficient variation, relative mean difference. These are the examples of the dispersion measurements. Okay, and another topic is measure of central tendency. This central tendency is used to represent the entire series. The property of concentration of value around a central value. The concentration of the property value of a central value is known as central tendency. This central tendency is divided into two types. They are mathematical and positional. In mathematical central tendency, it contains arithmetic mean, geometric, and harmonic mean. And this arithmetic mean central tendency is again divided into two types. They are simple arithmetic mean, weighted arithmetic mean. This is about the measure of central tendency. When we move to the next topic, which is measure of dispersion, we already studied about the dispersion, which is the data. Dispersion of data is degree to which numerical data approached to spread the average value. These dispersion types are biological, real, and experimental. And in this measure of dispersion, a small topic which is range. The range of a set of data is the difference between largest and smallest values are classified by the range. However, in descriptive statistics, the concept of range has a more complex meaning. That is, the range is the size of smallest interval which contains the all data and provides the indication of statistical dispersion. This range is measured in the same units as the data. Now, next small subtopic is correlation. The relationship between two continuous variables is defined as a correlation. This correlation is divided into positive, negative, zero correlation, and second one is linear, non-linear correlation. Third one is simple, partial, multiple correlation. These are the three different types of the correlations. In these correlations, we can observe the different types of graphs which are plotted by the x and y axis. Thanks for watching. In this class, I am going to discuss about the ANOVA. ANOVA means analysis of variance. The analysis of variance is similar to the regression analysis. It is developed by the R.A. Fisher. ANOVA is widely used by the researchers to investigate the underlying relationship between the dependent variable and independent variable. It is a statistical tool which is used to analyze the variations between data and inequality population mean. This ANOVA is an analysis of variance which is similar to the regression analysis, and it assumes the population distribution which is normal and samples are random and independent. The homogeneity of sample variance is used for the t-test to find the equality of mean and relative variance. This ANOVA is directly proportional to the variance between sample means divided by variance within the sample. And these ANOVA techniques are used in the several types. They are variants which occur due to one variable. In this case, the change occur in a variable because of only single variable. And second one is variance which occur due to two variable. In this case, the impact will be occurred by the two variables. ANOVA test types. Types of ANOVA test. Types of ANOVA test includes one-way ANOVA and two-way ANOVA. In the one-way ANOVA, the one-way ANOVA has one categorical independent variable and normally distributed continuous dependent variable. The independent variable divided into two or more levels. And in this one-way ANOVA test, this test is used for the difference in means of dependent variable, which is broken down by the levels of independent variable. And when we talk about the two-way ANOVA, in this two-way ANOVA, more categorical independent variables are occurred. The independent variable which is divided into two or more exclusive groups. Welcome to the HEMA pharmacy. In this class, we are going to discuss about the sampling. This sampling is the process which fixed number of observations are taken randomly from a large population. This is the technique which is fundamental for the behavioral research. It is impracticable and impossible to study the whole population due to the practical limitation and the factors that are indispersible and operative in this studying population. For the same reason, making research findings economical and conceptual of sampling has been introduced. The important objective of the sampling is to obtain the maximum information of the population under study using the money, labor and time, which is mostly this sampling method is majorly used to find the minimum information about the minimum to maximum information about the population by using money, labor and time. This sample design is a definite plan which is obtain a sample from the sampling frame to design. And this method or this technique which is adopted by the researcher in selecting the units of sampling from the population, which is called as sampling design. Some good characteristics of the sample design. This sample design must result in a truly representative sample and it is contain small sampling errors. These sampling design must be available in the context in funds. The sample design must be systematic bias and which is used to study the applied in general for the universe. And another subtopic is population. The population or universe is the sum of total objects under the study. The numerical details of collection of individual qualities of the results, which is known as universe. Every particular thing which is from the microorganism to the big animals and humans, that all the things which are included in the world is called as universe. This universe is classified into two types. They are finite universe and infinite universe. In the finite universe, the height of the students in a particular class, this is the best example for the finite universe. And infinite universe, the number of infinite number of members who are included in the universe. Means, for example, pressure of various points. When we take in a room which is a classroom, the temperature can be increased and decreased, or the pressure will be also not finite. We cannot say this is the exact pressure. So that's why the infinite universe. And sample is a part of population which is